Good afternoon, everyone. I hope you enjoyed so far the NACF conference. My name is Ines Pankonin. I'm a researcher from the University of Lisbon in Portugal. This is Gegay Lukács from Montreal, McGill University in Montreal. And we have the honor today to serve as a chair in the session about CFTR structure, function, and cellular biology. These are our learning objectives. So we will hear about new or novel structural insights on CFTR folding, but also about the complex activation mechanism of CFTR by the protein kinase A. We will hear about novel approaches tackling CFTR or rare CFTR genotypes, but also novel results on the outcome or the impact of the CF whole genome sequencing regarding the CF pathophysiology. We will have 4 speakers today. Every speaker will have 20 minutes, and this is followed by a 5 minutes question and answer discussion. And so the first speaker, we will have John Hunt from the Columbia University. He will talk about the Cryo-EM studies on human CFTR unfolding. The second speaker, we have Laszlo Xanadi from the Semmelweis University in Budapest, and he will tell us novel insights about the molecular mechanism of the CFTR channel regulation or CFTR activation by the protein kinase A. The third speaker will be Catherine Oliver from Emory University School of Medicine, and she will talk about ribosome-directed small molecules and nucleotide-based approaches to overcome distinct subclasses of CFTR variants. And the last speaker will be Gary Cutton from Johns Hopkins University, and he will tell us about the CF Genome Project, which informs about CF pathophysiology. And with no further ado, I would like to ask John to come here. Let me just see. Start. Do you want the laser point?
Yeah, that's good.
Okay.
Yeah, I think we're all set.
Hmm?
Yeah, it's good.
No, but you want the laser pointer?
Oh, yes. If on the screen, you mean?
On the screen,
yes,
that
that
would
would
be.
be.
Also use that one. Okay.
Oh, can I do it on the screen?
Laser pointer. Okay, we have to. It's not working. This is the pen. This is the logo.
Yes.
Now
Yes.
we have it.
Okay. Okay. That's good. That works. Good. Thank you. I'd like to thank the organizers for giving me the opportunity to present to you today, and I'd like to thank the CFF for supporting the related research in my lab for the last 25 years. It has been an interesting ride and an opportunity to do some things that I probably would have been very hard to get funding for from any other source. And I'll also say I totally appreciate their dedication to the cause and what they've done for people with the disease. It's an honor to work with the CFF for all this time period. So these are the people who have contributed to the work I'm going to be telling you about today. I like to call them the heroes of this CFTR universe. Most of us have been working together since about 2002, when as part of something that was called the CFTR Structure Consortium. And it was during that time period that the constructs that we're working with were developed, and also that the NBD1 folding studies that I'm going to summarize very quickly were done as part of that consortium. And so the key contributors today in my lab were Chi Wang and Blaine Laughlin, Sergei Vorobiev, and Ollie Clark have also helped with EM. Gergely, our co-moderator, has been a key collaborator for the last 2 years. Bob Grashushi runs the EM facility. Joachim Frank is our key EM, who taught me, also show in a moment, how to do EM. Jack Reardon, Ina Urbash, Christy Brett, Wendy Yang, and John Kappas have all been involved in developing the protein constructs that we're using, and John and Wendy still produce the protein. And I should say, I neglected to put his picture here, but Andrei Alexandrov also contributed some electrophysiology data. Okay, so this is the slide explains sort of my introduction to Cryo-EM and the way that I think about it. My lab learned to do Cryo-EM from Joachim Frank, who shared the Nobel Prize for developing the computational methods that are used to do this. And the basic issue there is that individual images, although there's very high resolution given by electron microscopy images, because the wavelength of the electron is extremely small, the images are extremely noisy. And so that it creates mathematical challenges. It also creates conceptual challenges that essentially have taken the field a while to work out, and it's still in the process of being done. And what I'm going to show you today are sort of pushing the envelope, basically, on what you can do under the circumstances and what you can learn about proteins, which I hope you'll conclude is truly amazing. So this is my apartment is in New York City, is exactly 10 floors below this. And so this blaze was going on one night. Well, of course, we were basically up all night. The New York City Fire Department was amazing, but we were up all night. Just around the time I got to sleep, my wife comes bounding into our bedroom yelling, "Joachim won the prize, Joachim won the prize," because that was the night that the next morning Joachim's award of the Nobel Prize was announced. So this is the way I think of Cryo-EM, both because of this experience, but even more so because of what it enables us to learn about proteins. And so an overriding hypothesis of what I'm going to tell you about today is that static snapshots of proteins trapped in crystal lattices has seriously biased conceptual understanding of protein functions. So my lab studies 3 different ABC superfamily proteins. I won't tell you about the details of any of these things. Of course, CFTR I'll go into in detail. But we also study B2CDF and ABCF proteins, which are translation factors. So all of these proteins have the homologous ATPase domains. Their ATP-dependent mechanochemistry is rather similar. That's what my lab first started working on when I first formed the lab. And we've continued to study these 3 things now for a very long period of time. So although they have the homologous ATPase domains, their molecular mechanisms are very different. And there's commonality in the way they use ATP, and then they have very different molecular mechanisms. One thing that we have seen in the last couple of years is that dynamics are totally key to every one of these things, and they're key in different ways. And the dynamics that are really at the core of the function of every one of these things is really beyond the grasp of what you can get with crystallography, but we can start to get it with Cryo-EM. So an alternate title for my presentation might be Cryo-EM G-STIRs Reveal the Secret Lives of ABC Proteins. So what is a G-STIR? A G-STIR is a global conformational ensemble reconstruction. And what that means is when you take your sample, you try to find every conformation of your protein that is in the sample. And this is different from what now many people are now trying to do this. Pretty much everyone who's doing it is doing it a bit differently. But still, the bulk of the people doing Cryo-EM are essentially trying to find one conformation. So they're using the electron microscope as if it were an X-ray crystallography experiment to find one conformation. And I think they're throwing away like the richness of what you can really learn from it. So the global conformational ensemble reconstruction is an exhausted, but admittedly somewhat subjective search for all alternative conformations. And then this is all technical stuff that I don't think is relevant to this audience, but the most powerful reconstruction refinement methods, an important technical point, the most powerful reconstruction refinement methods can effectively hide alternative conformations. So there can be things hiding in your sample, in your sample and in your reconstruction math that are really very important and very interesting, and it's very easy to miss them unless you work at this. So the next slide, this isn't CFTR, but it summarizes a different project. But in this case, we put the entire Cryo-EM project in one figure. So I can just very quickly, not going to go through the details, but just to quickly outline for you what it is we're trying to do. So the first step in doing a Cryo-EM reconstruction is getting 2D classes. These are essentially just aligning, rotating the different particles so they sort of look the same in projection. And then from there, you try to reconstruct a 3-dimensional structure. Well, again, what most people do in this point, because some of the particles are actually just bad and you do need to throw them away. But also there's just a huge amount of conformational heterogeneity. And so people are usually trying to find the structure. And so what they do typically is they just throw away most of the data and they try to find some small subset of the particles that are in one conformation. And so most published Cryo-EM structures are using somewhere between 3 to 10% of the particles, and they're throwing away all the rest of the information. So what we've done is rather different. After finding all the conformations through an exhaustive, somewhat manual search process, we go back to all the particles, and then we use basically a reference space classification with all these conformations, and we basically both classify and clean the particle stack by iteratively assigning the particles to the major conformations. And when we do this, we also have a number of dummy volumes that are just sort of junk things. And so the junk particles slowly get peeled off by these dummy volumes, and then the particles that really match the CFTR, or in this case something called SCR, get passed through after every cycle. And this is a plot that shows, in this case, we had 3 distinct conformations, and you can see the relative population of the 3 distinct conformations is different, is equivalent throughout the cycle. And so in this way, we capture the complete conformational ensemble that we can find in the sample. And in this case, this is what we learned from it. So this is a movie showing a transition, a density transition between the 2 major classes. This ends up being an NADPH-dependent enzyme. It's a superfamily with 4 million new unique sequences in the sequence databases. There's 78 structures of this thing, of 30% ID homologs in the PDB. So there's a huge number of these, and have crystal structure determined. Every one of these things is a symmetrical tetramer in the PDB, but when we look in solution, we find 25% of the population in this one is in an alternate conformational state. In that conformational state, the domains have essentially peeled apart. When they peel apart, the active site becomes disordered. And essentially what's going on here is these subunit rotations are dynamically disordering the active site, and they are kicking off the substrate. And so, the products rather. And so here's a critical element of dynamics for the function of this enzyme. And we have all sorts of other biophysical data that show this, that you just miss if you do crystallography or if you do cryo-EM, where you're just trying to find one structure. So so fluctuations in intersubunit interactions drive active site unfolding. So this is another example. This is from the ABCF protein project in my lab. This is the catalytic core of the ribosome. The ribosome is believed not to actually stabilize the transition state for peptide bond synthesis. It's believed to catalyze things just by locking them in exactly the right place to react. And so it's a lock and key kind of mechanism. But of course, once you make a peptide bond, you have to extrude the polypeptide from this exit portal here. And if it's locked down, you can't do that. So what we've found is that hiding in all cryo-EM data sets that people deposit in the databases, there's actually a disordered state of the catalytic core of the ribosome that is probably also key to its function, is probably key to letting the substrate, letting the nascent chain move on between the cycles where the lock and key, right? So another example where hidden dynamics are probably at the core of what makes this thing work. So on to CFTR. So I think that, to my mind, one of the really exciting things about cryo-EM is that I think it's going to allow us to sort of link what I call the cell biology worldview, gel bands, fluorescence microscopy, schematic diagrams, to the molecular biophysics worldview, which involves coupled conformational equilibria, free energy, and atomic coordinates. I'll give you an example of that in a couple of minutes. And so where we are now is people are beginning to develop cryo-EM methods, approaches, and just play with things so that they can actually get conformational dynamics from silico particle reconstructions. So I'm going to show you examples of that today. And then the next step, which we're not there yet, is doing the same thing in situ in cells using cryo-electron tomography. So I think we're not here yet, but we're in the middle of this revolution now. And combined with AlphaFold, this is just an incredible opening of the horizon in terms of protein structure. And in the next few years, we're going to be able to understand protein structure, function, folding in a way that I think it really is revolutionary compared to what we know now. All right, so before going to showing you CFTR structures, I'll just point out that correctors work during a 45-minute time period after the, and Gerge has data, very nice data showing this, the times when correctors act on CFTR is following synthesis of the full chain. That's where you get most of the corrector activity. And so that's a 45-minute period, roughly, where very complicated things are going on. What we are doing is, of course, using cells to make it in tissue culture bioreactors, and then we're purifying it in weird buffers at 4 degrees. And then we think we're learning something about what's going on in the cell. And I do think that's true, but this is something that is difficult, and a caveat is, right, I'm going to tell you hypotheses, and I think additional work is going to be needed to figure out which of the hypotheses, well, how, exactly how they relate to what goes on in this very complicated in vivo biogenesis process. So now I'm going to introduce you to CFTR structure, so you'll need to see these, understand a bit about this, and understand the rest of the talk. I'm also going to use the same coloring throughout, so I'm now going to slow down slightly and explain to you the coloring and the structure. So there are 2 nucleotide binding domains, NBD1 and NBD2. ATP binds at their mutual interface. The main thing that ATP binding does is pull this interface together from a more expanded state. I'll show you later. The F508del mutation is at the interface between NBD1 and intracellular loop 4. In addition to the NBDs, there are, of course, two transmembrane domains. The transmembrane domains are domain swapped. So there's sequence transmembrane domain at the N-terminus, I've shaded here in different colors of blue, and the the C-terminal transmembrane domain I've shaded in colored in shades of green. But two of the helices from the N-terminal sequence domain basically form part of the C-terminal structural domain and vice versa. So there are two alpha helices out of the 6 that span the membrane that are domain swapped between the two sequence domains. So the coloring that I've used is based on the sequence domains, not the structural domains. The R region, of course, is disordered, so we don't see it in the, it is present in all our structures. We see little snippets of it here. I can tell you about that after the talk if anyone's interesting, but we generally don't see it. At the N-terminus, there's a unique feature of CFTR called the lasso, and the lasso is totally important. I'm going to tell you a lot about that. And so, again, but a key point is the F508del mutation is at the interface between the MBD and ICL4 in that intracellular loop 4, which is coming from the domain swapped region of the second transmembrane domain. The ICL4 interacts very tightly with part of the lasso, this N-terminal extension. And the VX445 binding site we have found is basically at the essentially the helices that form ICL4 and the lasso form the proximal part of that to the site. The membrane embedded region of these helices proximal to the cytosol in combination with the lasso form the VX445 binding site. So so a key point historically is that stabilizing HNBD1 folding is necessary and sufficient to create the F508del biogenesis defect. So this is an essential starting point for understanding what's going on. I see people in the room who contributed to this basic understanding over the course of a decade. This was already known a decade ago. And so I'm not going to go into the details now, but I can, afterwards I'm happy to tell you, it's absolutely clear that to correct the folding, you need to correct the folding of NBD1. And the problem that immediately raises is that I'm not going to talk about VX809 today, but I'm happy to talk about it afterwards, the talk later. I'm going to focus on VX445, but VX809 binds at a relatively similar position down here near the lasso. Oops. That scared me. I thought that was the 3-minute warning. Anyway, so this is 50 angstroms, right? The corrector site is 50 angstroms away, right? And so that's, you've got to explain that. If you really understand the mechanism, you've got to explain that. So they have to act via an allosteric mechanism that's thermodynamically stable as NBD1. So I'm going to explain that in the next slide. Oh, and just one point I'll talk about later. The extracellular exit portal for the protein is back here down on the other side, sort of behind, and according to this view. And I'll say tomorrow I have another talk. I don't know how that happened, but I have a talk today's talk is on folding. Tomorrow's talk is on gating. So I'm not really going to say much about gating today. Okay, so I'm not going to go into this at all, but just to make the point that the correctors and potentiators were isolated by empirical screening. This is sort of a flow chart showing what that went. And so everyone in this room probably knows that, so I'm not going to go into this in detail here. But the point I want to make was I'm going to show you now the allosteric mechanism of action. This never could have been anticipated in advance of having the drugs and seeing how they act, basically. And so really the empirical screening was the essential approach. These things could not have, now we can understand them, they could not have been designed rationally. Okay, so this shows the key mechanistic features that are involved in the correction process and, importantly, the folding process. So this is what we call the V-shaped conformation of CFTR. The MBDs are pulled apart because, in this case, ATP is not holding them together in the more narrow conformation I showed you earlier. What I'm going to show you in a moment is that there's a dynamic equilibrium where NBD1 detaches from the transmembrane domain, and then when it detaches, ICL4 and the N-terminus lasso unfold. So basically, the binding of NBD1 is cooperatively coupled to the unfolding of the lasso and ICL4, right? And so the correction process has to recover NBD1, right? When F508del NBD1 unfolds, it is extremely aggregation prone. It is rapidly targeted for degradation, probably based on some aspect detecting its aggregation, whatever, something about its aggregation prone tendencies. Comparable mutations in NBD2 don't really do anything. There's something about the unfolding of NBD1 that causes the degradation, and reversing the unfolding defect is necessary and sufficient to to correct the disease defect. So how does that work? Well, so the unfolding transition that we've seen, I think our hypothesis is it explains it because of the cooperative coupling of the NBD1 detachment to the unfolding of the ICL4 in the lasso. If you refold the ICL4 in the lasso, you will then by mass action, essentially pull the unfolding of the NBD back towards the native state. And so that's sort of schematized here. I'm going to blow up. It's the same image, but just zoomed in. Right. So here's the state with NBD1 detached and the ICL4 and the N-terminus lasso all unfolded. When VX445 binds, it stabilizes directly the lasso. The lasso stabilizes ICL4. Once you stabilize that whole structure, that promotes the rebinding of NBD1. And so that's the model that we have of how this works. And so the key point is that by stabilizing the lasso, which stabilizes ICL4, which binds NBD1, that pulls the whole process back towards the native state, away from the unfolded state. And that's how you can explain the allosteric action of the folding of the V4X445 folding corrector. I'd have to say the 809 is probably more complicated. I don't have time for that today, but I'm happy to discuss it afterwards. Okay, so now I'm actually going to show you a bit of data. This is an example of a global conformational ensemble reconstruction. This is wild-type CFTR. This protein is absolutely wild-type CFTR. It has a couple extra residues at each terminus. Other than that, it's absolutely wild-type CFTR. This is our global conformational ensemble reconstruction. We have 3 conformations. They're very stable through the classification process. This V-shaped conformation is 40% of the population. Then we have what I'm going to explain a bit more in a moment. We call it NBD1-less, so that the NBD1 is detached, and then PU, partially unfolded. So these things are both lacking NBD1. You can see no density for NBD1, and they're lacking ICL4, and they're lacking the N-terminus of the lasso. Where they differ is that in the membrane-embedded part of TM11, in one of these samples, this is perfectly folded, and in one of these samples, it's gone. This is the kind of resolution of information we can get with Cryo-EM when you do it very carefully. It's not easy to get this, but we can get it very reliably when you want to. So key points here, again, this is wild-type CFTR, PK phosphorylated wild-type CR. NBD1 is significantly dissociated from PK phosphorylated wild-type CFTR just at baseline. So the delta G for this coupled folding binding reaction is about 0. The folding of ICL4 and the N-terminus lasso are cooperatively coupled to HNBD1 binding. I explained that to you a moment ago, and now this is showing part of the data. The membrane region of TM11 is also unfolded in some partially unfolded molecules in which ICL4 is unfolded. We're hypothesizing that this is a purification artifact, but that this is the essential species that's actually present all the time in PK phosphorylated CFTR, and it is accounting for it can explain the corrector activity. And so another thing we learned from this is the digitonin CHS, which is cholesterol hemisuccinate, which is running the structures. This impedes the conformational change forming the internal chloride channel, because in this case, we threw in 15 millimolar magnesium pyrophosphate. That locks the channel open in membranes. There's no even channel formation in digitonin. So digitonin, where we're doing this, does bias the protein away from the open channel state. Okay, so what that tells us also is that there's an insignificant population present in solution that has the ICL folded, but it does not have the NBD1 bound, and that just emphasizes that there is that the binding of NBD1 and the unfolding lasso are cooperatively coupled, and it indicates that there is pretty much an exact balance in the binding energy at that interface and the folding energy of ICL4 in the lasso. And I won't go through the details here. This is Gergely's paper that just came out. This is beautiful work in what he used HDX, and with the hydrogen deuterium exchange in solution, he showed exactly the same thing using mutants and correctors. He showed that this ICL4 is dynamically destabilized by the F508del mutation and other things that prevent biogenesis. Okay, so where am I? Okay, I'm going to go on for like 3 minutes. I pressed my luck a bit. Okay, so the ICL4 NBD1 interface is rigid. So what I'm doing here is I'm showing an alignment based on, here is aligned based on the interacting region of NBD1. And in this case, one structure is in solid colors and the other in semi-transparent colors. And when I align based on NBD1, you can see that this is all just one thing. These are all pretty much exactly the same. That means this interface is very rigid. Although the delta G for its formation is about 0, once it's formed is very rigid. Presumably the entropy of the unfolding is being balanced against the enthalpy of the interface formation. If I align instead on the core of TMD1, right now you see something different. You actually see that this is all right over here. When you align here, everything over here is blurred out. When we align on TMD1, this whole region also becomes blurred out. And so what this is saying, and then these are other views based on this TMD1 alignment. And so this is probably the most interesting one, right? So here, these are a bunch of structures, a bunch of very different conformations. The blue is the sequence membrane spanning domain 1. You can see that in this alignment, these things are all very, very similar. But now what's blurred out is TMD2. And so essentially, this rigid handle, as it were, is propagating down through conformational changes that are then being propagated through TM10, TM11, and that are controlling the conformation down here in the exit portal region. And I'll show this. I'm going to talk a lot more about this tomorrow. So this is zooming in on the exit portal region. And what you see here is basically in all of although there's sort of a continuous range of conformations in the totality of the transmembrane domain, in the exit portal region, there are only 2 distinct conformations. And so what we get from this is that the rigid displacements of the HNBD1 and ICL4, they drive a continuous deformation of transmembrane helices 11 to 12. I'm sorry, that's a typo. It should say 10 to 11. Sorry about that. And this controls a discrete conformational transition in the exit portal, and I'll talk more about this tomorrow. And so sorry, something went wrong there. Yeah, so this is illustrating the main conformational transition that's involved in opening the channel, and that'll go into more tomorrow. Okay, so for the moment, just to summarize this is essentially this is just blowing up the front side view of this. Normal allostery involves the NBDs being pulled together by ATP. That propagates an allosteric conformational change down to the exit portal. I'm going to show you in a moment now that what happens with VX445, because it's also a potentiator, it is actually acting in inverted allostery. Using inverted allostery, it is manipulating the conformation of the transmembrane domains, and that's pushing the TMD around. So I'm sorry, that's pushing the NBDs around. So I'm going to skip. Well, I'll just show this quickly. We, at this point, have about 100 different structures in 15 different conformations. Some of these things we think are probably purification artifacts, because we can see so much now. We can see the protein falling apart while we're purifying it. And then some of them, however, we think are physiologically vital things. And however, distinguishing between the two is going to require additional experimentation. Is definitely one of the challenges for the field. So I'm going to show just one more slide before I wrap up. So one of the things that we've seen is we get structures where the exit portal is unfolded. And we get a varying amount of this in different samples. This is the exit portal region that I showed you a moment ago is down here in this view of it, right? So that's the exit portal right behind that helix. That's the N-terminus of helix 8. So what you can see here, if you look even just very vaguely, this is actually the VX770 binding site. The exit portal is down here. You can see that they're just dramatically different conformations in here. And it's very clear that the exit portal has just entirely unfolded in many of the molecules that we have in solution. I think this is the last panel I'll bring up. And so when we have solved the structures, there's always, this is a very weird piece of protein structure here. There's actually a break in the transmembrane helix in the middle of the membrane, which will expose the very polar N-terminus of the helix of the membrane. This is basically never seen in other structures. VX44770 binds in exactly that site. I'll talk a little bit about how VX770 tomorrow. Basically, we have no idea how it works. It does not do anything at all in digitonin. But next to this, there's always a weird density blob. And in our best structures, it looks as if that density blob is actually a phosphorylserine molecule. So what it looks like has happened, that there's been a phosphorylserine molecule where the head group is actually flipped up in the middle of the membrane. That is stabilizing this break in the helix and actually forming part of the VX770 binding site. So this is the kind of incredible detail that we can now get out of Cryo-EM structures, where we can even begin to see local unfolding. Our hypothesis is this comes from loss of phosphorylserine during purification, that it's not physiological, but that's a hypothesis, and obviously there's more work to be done. So with that, I will finish. Yep. And I think I've used up my question time.
Thank you, John. So Thanks for the dense talk and the fuzzy picture sometimes. But I think we have time for 2 quick questions.
I wish I could listen to it in slow motion, but
Well, we could do that. We could do that over drinks.
anyway, the question regarded the VX445. You mentioned lasso and ICL4 for folding, and you actually had it written potentiation. Is it the same? You think it's
So
the same?
potentiation, I'll talk about potentiation tomorrow.
Okay.
So yeah, well, now here, very quickly, without going through any detail. No, here, this shows that mutations have a different effect on correction and potentiation. So they're related, but the key there is that TM10 and 11, they are like the allosteric linchpin for the whole system, and they're binding in the membrane part of TM10 and 11. Just by binding there, it will stabilize the lasso, and that's probably enough for correction. To get the potentiation, you need to do the right thing to TM10 and 11, and it happens to do that. And I think that's why you see people now have lots of compounds that bind in that site. Some of them potentiate, some of them do not. Mutations in the site Gerge showed, some of them block the potentiation and not the correction. Yeah, so it's the same region, but because the allostery is a bit tricky, it's not exactly the same effect. Is that?
I think my question follows up on this almost exactly. Your 2 arrows, normal allostery and inverted allostery, are running right parallel to each other. So if 445 is interfering with one direction of the arrow, does it?
Yeah, so tomorrow I'm going to talk about gating. I'll go into this in more detail. But basically, so what usually is happening is that ATP is pulling the NBDs together, right? And when you pull the MTVs together, this segment of it is rigid, as I showed you, but this undergoes a continuous deformation. So as the NBDs pull on this handle of ICL4, they produce a continuous deformation in the lower part here. At some point, it clicks between two different conformations way down here in the exit portal.
But essentially, that links correction and potentiation.
Because this corrector site is also at the allosteric heart, right? So usually MBDs are pulling these things together, and then the transmembrane helix. What VX445 is doing is pushing the transmembrane helices into this conformation. And the way we know that is because in G551D, I'm not showing you those structures, but in G551D, you can see it's sort of pushing the NBDs close to one another, but they don't actually encapsulate the ATP. Because the mutation is right there at the ATP binding site, they push them towards one another. They don't actually encapsulate the ATP. So that's how they get the inverted allostery. G5, sorry. They what? Oh, is that right? Well, I can probably with ADP. Well, let's talk about that. Yes. Yeah. Yeah. Thank you.
We've got one more question here for you. I have to read it out loud. It's from Andrew Kleist. I love your asking questions about dynamics with EM. Have you or others been able to correlate EM and ensembles with ensemble data from other methods like SM-FRET?
So for our project on B2CD, we've used primarily SM-FRET. SM-FRET is very powerful, but it does not have high spatial resolution. What gives you high spatial resolution is what Gerge did. The Gerge's HDX gives you residue by residue information on dynamics as illustrated by H-hydrogen deuterium exchange. You can also do some things with molecular dynamics. So FRET is tremendously powerful. It does not have the spatial resolution to really. You could sort of get a crude match. It does not give you the kind of spatial resolution you could use to check the details.
Thanks very much, John. So our next speaker is László Csanádi from Budapest.
So first of all, I'd like to thank Gergely and Ines for their kind invitation. It is really a great honor to be speaking at this meeting. And so today I'm going to summarize some of our recent work trying to understand the mechanism by which protein kinase A, or PKA, regulates CFTR. So as you all know, PKA activates CFTR by phosphorylating about a dozen target serines in the R domain, the regulatory domain, and phosphorylated channels are then gated by ATP binding at the two nucleotide binding domains. And the mechanism of action of ATPs is relatively well understood. Upon ATP binding, the two nucleotide binding domains form a tight dimer, and this NBD dimerization opens the channel pore, and then following hydrolysis of one of the two bound ATP molecules, the dimer loosens, and that is coupled to pore closure. But in living cells, the channels are bathed in saturating ATP all the time. So ATP cannot be considered the regulator of the channel. It is more like a fuel, which is required to drive gating. But regulation is achieved through R-domain phosphorylation by pKa. And so despite its clear physiological relevance, the mechanism by which this process happens is little understood. Apart from the fact that the unphosphorylated R-domain is clearly inhibitory because its deletion results in channels that can gate readily in the presence of ATP. And so today I'm going to try to summarize some of our recent progress trying to understand the mechanism of this regulation. So a first mechanistic insight came from recent Cryo-EM structures solved in Zhu Chen's laboratory of unphosphorylated closed and phosphorylated open CFTR. In the unphosphorylated channel, the R-domain is seen wedged in between the two NBDs, preventing their dimerization. And upon phosphorylation, it is released from this inhibitory wedged in position and likely becomes entirely disordered. Now, from the structures, it was not clear how the pKa can have access to such an occluded R-domain. And so at the time, we proposed that perhaps infrequently, even the unphosphorylated R-domain is released from this wedged-in position, and that is when the pKa can grab and phosphorylate it. And so such a two-step activation mechanism also seemed consistent with our observation that in inside-out patches upon pKa exposure, macroscopic CFTR currents do not activate following a single exponential time course. Instead, the activation time course is clearly sigmoidal. And such a sigmoidal time course can be well fitted by a model which assumes that the channel has to shuttle through two non-conducting states before it opens. And so we propose that perhaps the first of these two steps reflects this slow or infrequent spontaneous release of the unphosphorylated R-domain, resulting in channels which can then open, but with very small open probability. And if we zoom in on this segment of the trace, you can indeed see that even unphosphorylated channels do open very infrequently in the presence of ATP. But then the real activation would be caused by phosphorylation. And so another robust phenomenon that you can see here is a very rapid partial deactivation just seconds after pKa removal. And for a long time, we interpreted this to reflect rapid dephosphorylation of a subset of the phosphoserines by membrane-associated endogenous phosphatases, resulting in partially phosphorylated channels that can now gate at a lower open probability. But this interpretation raised several questions, and in particular, an alternative interpretation could be that in addition to phosphorylation, mere binding of the pKa could stimulate the channels, and it could be this component of the stimulation which is lost immediately upon pKa washout. So how can one dissect effects of pKa binding versus phosphorylation? And we thought of two approaches. One would be to use an ATP analog that can gate CFTR but cannot bind to pKa and can therefore not be used for phosphotransfer. And another approach would be to generate mutant CFTR channels in which all the pKa target serines are removed. So we started with this first approach and indeed managed to find a suitable ATP analog. Phenylethyl ATP or PATP was well known to gate CFTR channels efficiently. This was done in T.C. Huang's lab. But we also found reports in the literature that PATP cannot bind to pKa and cannot be used for phosphotransfer. So can pKa still stimulate channels that are gating in the presence of PATP? So what you can see here again is channel activation by pKa in the presence of ATP and partial deactivation upon pKa removal. But if we now reopen these phosphorylated channels by PATP, second application of PKA again results in reversible additional stimulation. And this cannot be due to phosphorylation if the PKA is indeed unable to use PATP for phosphorylation. And what was even more surprising is that even this tiny activity of unphosphorylated channels in the presence of PATP can be robustly stimulated by PKA exposure. However, again, in contrast to the effect of PKA in ATP, which leaves a lasting effect that must reflect phosphorylation, the effect of PKA in the presence of PATP is again completely reversible. So we also verified whether PKA is indeed unable to use PATP for phosphotransfer, and for this we used chemtide, a soluble substrate peptide of PKA. The mobility of chemtide on thin layer chromatography sheets is reduced upon phosphorylation, and that allows us to monitor and quantitate by densitometry the rate of chemtide phosphorylation. Here you can see that in the presence of PATP, there's virtually no phosphorylation. And so for the conditions used in these electrophysiological experiments, we calculated the difference between phosphotransfer rates in the presence of PATP and ATP is 10,000-fold. In contrast, if we compare the activation rates by PKA in the presence of PATP and ATP quantified through activation half-times, those are virtually identical, and that precludes the possibility that this activation in PATP could be due to phosphorylation. So in summary, in addition to phosphorylation, mere binding of PKA causes reversible stimulation, and these two stimulations are additive. So as an alternative approach, we also generated mutant CFTR channels in which all 11 confirmed PKA consensus serines were mutated to alanines. And this 11SA construct remained strongly activatable by PKA. In fact, that had already been documented in early studies and was thought to reflect phosphorylation of some as yet unidentified target serines. But what was not observed previously is that this effect of PKA on the mutant is again completely reversible, in contrast to the effect on wild-type CFTR, which in which case there is a lasting effect, as you have seen. And so the response of the mutant can be explained by reversible activation through PKA binding. So based on these data, we constructed a simple model in which the R domain exists at a rapid equilibrium between wedged-in and released conformations. That equilibrium is shifted towards wedged-in for the unphosphorylated R domain, but is shifted towards released for the phosphorylated R domain. The wedged-in conformations are inactive, but the released conformations are all completely active in the sense that they are able to gate in the presence of a suitable nucleotide. And regardless of whether the R domain is phosphorylated or not, PKA binding can further shift this R domain conformational equilibrium towards released by binding the R domain and preventing it from returning into the wedged-in position. And this simplified model can quite well describe even such complex experimental time courses that I've shown you before. So next we are interested to understand the mechanism by which PKA can prevent the R domain from returning into the wedged-in position. So is this done as suggested in this cartoon by a direct interaction between the PKA substrate binding site and some R domain loop, kind of grabbing the R domain and preventing it from returning into the wedged-in position? Or is it perhaps through some kind of an allosteric effect that is mediated by some other part of the kinase? And so to address that, we used the PKI peptide, which is an analog of a substrate peptide in which the target serine has been mutated to an alanine. And so PKI is a high affinity competitive inhibitor. Can it also prevent reversible CFTR stimulation? And you can see here that PKI can not only prevent irreversible activation, that is phosphorylation as expected, but reapplication of PKI following phosphorylation can essentially entirely abolish this reversible component of CFTR activation. And the same is true for the reversible activation of unphosphorylated channels gating in PATP that can again be prevented by PKI. So it appears that reversible activation is mediated through a part of the PKA, which is also involved in substrate binding. And so a simple interpretation would be that the substrate binding region just binds some part of an R-domain loop and thereby prevents it from returning into the wedged-in position. Now, we also uncovered another important structural determinant of the PKA-CFTR interaction. All the experiments I've shown you so far were done on native bovine PKA, purified from bovine heart. I will denote this PKA prep BPKA henceforth. But in order to be able to structurally manipulate the kinase to introduce mutations into the kinase, we also expressed the bovine PKA in E. coli and affinity purified it, and that prep, I will call it recombinant PKA or rPKA. And so the recombinant PKA proved fully active on the soluble substrate chemtide, but when we tested it in inside-out patches, we found that the recombinant enzyme can only very slowly activate CFTR channels, and the maximal activity achieved by the recombinant protein remained much smaller than what could be then elicited by the native bovine PKA. And that inefficiency of the recombinant protein is partly due to a complete lack of reversible stimulation, but even the irreversible component fails to reach a maximal value. So this absence of reversible stimulation is again also seen for unphosphorylated channels gating in PATP. Now, this inefficiency of the recombinant PKA is even more evident on the delta F508 CFTR channel, for which the recombinant enzyme activates only about 10% of the maximally activatable current. And because nowadays most people carrying delta F508 mutations receive pharmacotherapy, we also compared the efficiencies of these two PKA isoforms in the presence of the potentiator VX770. And you can see that even in the presence of the potentiator, the recombinant enzyme activates only about half of the current that can be activated by the native bovine PKA. And so due to its potential clinical relevance, we were interested to understand the reason for this difference between the two PKA isoforms. So the amino acid sequences are identical, but their post-translational modification patterns are different. And one difference is that the native enzyme is N-myristoylated, whereas the bacterially expressed protein is not. And due to the presence of this N-myristoyl group, the native bovine PKA is able to readily associate with membranes. So we wanted to understand whether that difference might account for the different efficiencies towards CFTR. So there is a bacterial cysteine protease, EPA-J, which is able to cleave off the N-terminal myristoyl glycine from myristoylated proteins. And so we expressed and purified the EPA-J and first tested it on soluble peptides. We had two versions of the N-terminal peptide, the first 12 amino acids of the bovine PKA enzyme synthesized, a myristoylated and a non-myristoylated form. The two peptides can be readily separated on thin layer chromatography sheets because the myristoyl peptide has a much higher mobility. And you can see that in vitro treatment of this peptide, the myr peptide with EPA-J readily results in cleavage of the N-myristoyl group. So we then incubated full-length native bovine PKA enzyme with EPA-J and then again purified the PKA from the mixture. And this EPA-J treatment did not affect the activity of the bovine PKA towards soluble chemtide, but then when tested in inside-out patches, we found that EPA-J treated bovine PKA can again activate CFTR currents only very slowly, about fourfold slower than the untreated enzyme, so about equally fast as the recombinant enzyme. And you can also see that EPA-J treatment completely eliminates the ability to reversibly activate the CFTR channel. So we conclude that demyristoylation does not affect the phosphotransfer rate of pKa towards soluble substrates, but it greatly slows CFTR activation kinetics, and it eliminates reversible CFTR stimulation. So can we also reverse this pKa modification? Now, in the cell, myristoylation is done co-translationally by the enzyme N-myristoyl transferase, or NMT. And indeed, the purified NMT enzyme could myristoylate our N-terminal pKa peptide in vitro. But due to solubility problems, we were unable to in vitro myristoylate the full-length recombinant pKa. So instead, we co-expressed NMT with the pKa in E. coli and then purified the pKa from these cells. And again, this NMT co-expression did not affect the phosphotransfer rate of the resulting enzyme towards soluble chemtide, but it did greatly accelerate CFTR current activation kinetics. Although we were unable to fully recapitulate the efficiency of the native protein, so clearly even when NMT is co-expressed with the pKa in the bacteria, the myristoylation stoichiometry of the resulting pKa remains far from 100%. So in summary, we have identified two important structural determinants of the CFTR-pKa interaction, a direct interaction between the pKa substrate binding region and CFTR and the N-terminal myristoyl group. Both are required on one hand for rapid CFTR phosphorylation and on the other hand for reversible CFTR activation by pKa binding. Blocking the substrate binding site with a competitive inhibitor peptide eliminates both phosphorylation and reversible activation, and splitting off the N-myristoyl group greatly slows CFTR phosphorylation and again eliminates reversible activation. And the final thought, given that N-myristoylation occurs post-translationally in living cells, does this finding have any physiological or clinical relevance? Well, until recently, N-myristoylation was believed to be an irreversible modification, but the recent discovery of IPA-J showed that that is not necessarily the case. So what kind of protein is IPA-J? IPA-J is a virulence factor, which is secreted by the bacteria directly into the cytosol of the host cell through a type 3 secretion system. And of course, IPA-J is a Shigella protein, and Shigella is not a strain that is relevant for cystic fibrosis, but Pseudomonas, Burkholderia, and Stenotrophomonas strains, the bacterial genera that commonly infect CF patients, they also use type 3, 4, and 6 secretion systems or outer membrane vesicles, depending on the stage of the infection, to directly deliver virulence factors into the cytosols of epithelial cells. And the blast search through the genomes of these bacterial genera indeed uncovered a very large number of IPA-J homologues, cysteine proteases with uncharacterized function. And for one such Pseudomonas protein, we could indeed show that it is able to demyristoylate pKa in vitro. Now that, of course, doesn't mean that this would also happen in vivo, but if among these many uncharacterized virulence factors there are enzymes that are capable of demyristoylating pKa in vivo, such enzymes could contribute to the colonization of the CF lung at some stage of the infection, and so could be considered novel drug targets for the treatment of CF lung disease. And so with that, let me end by thanking the people who have done the work. Csaba Mihály has done all the electrophysiological recordings. Jordan Jordanov and András Szőlősi have done the protein biochemistry and the enzyme assays, and Beata Törőcsik helped in the construction of mutants. And I would also like to thank for the continued support through these funding agencies, especially the CF Foundation, and you for your attention.
Thank you, László. So, question, please.
So I have two questions. First, I assume that you're using the holo recombinant bacterial enzyme as opposed to the catalytic subunit. have you compared the
This is catalytic subunit.
It was just the catalytic
Just the
subunit.
catalytic subunit, of course.
I see. Okay. And then secondly, does the 11S mutation interfere with alter biogenesis at all? Did you look at that?
Probably to some extent. So we do get somewhat smaller expression levels for the mutant, yes. But we have not systematically pursued that.
Hi, great talk. Ian Thornell, University of Iowa. I was wondering what role protein phosphatases would play. Would they speed up the decay? Would they lower the plateau? Do they interact with the kinase activity?
So whether there are phosphatases in these patches is the question, or in an intact cell.
Or if you overexpress phosphatases, whether that would change the kinetics of that decay. That's
Yeah. So our conclusion was that in our inside-out patches, there are probably no membrane-associated phosphatases, because that rapid partial current decay is clearly not due to dephosphorylation, and the remaining current then is very stable over the time course of tens of minutes. So I think that in our cell membranes there are there's very little endogenous phosphatase activity. These are inside-out patches that are pulled away from the cell.
Yeah.
Yeah.
Hi. So really not an expert on this topic, but as far as I know, cyclic AMP also plays a role, right, in activation. And you,
I haven't talked about that.
No. So maybe just for me as a non-expert, where does it come in your story?
Yes, I should have mentioned that probably. So the PKA holoenzyme contains two types of subunits: two regulatory subunits and two catalytic subunits. So it's a tetramer, heterotetramer. And the holoenzyme is inactive. And when cyclic AMP levels rise, the regulatory subunits bind cyclic AMP, 2 molecules of cyclic AMP each, and that will then release the catalytic subunits, and then the free catalytic subunits are active and go and phosphorylate their targets. And so in our experiments, we have just used the purified catalytic subunit, which is active on its own.
Great talk, Lasso. I have one question about your experimental results, and that is, if there's no dephosphorylation, then how come in one of your recording, after you remove ATP and then apply PATP, you get smaller current? Yeah. And PATP actually gated CFTR better than ATP, right?
Yeah, but I think you know the answer for that. So that is because if you remove all nucleotide for a prolonged period of time, like 2 or 3 minutes in that experiment, then eventually the NBDs lose ATP, and the protein probably assumes this kind of V-shaped structure that John has shown before, and becomes inactive. And then when you reintroduce nucleotide, PATP or ATP, then you can immediately recover only the part of the population which has not undergone that kind of inactivation. And so you can see then that the current then continues to rise slowly over.
I thought about that possibility actually in the audience. And so that's why the next question is, have you done an experiment with shorter ATP washout?
Yes.
And then PATP activate more?
Of course, yes.
No more questions? Then maybe I have one quick affinity of the catalytic subunit of the phosphorylated versus non-phosphorylated R domain. Do you have any idea? And since we're having 10 phosphorylation sites, what's happening after the catalytic subunit binds? Does it have a chance to off, on, rephosphorylate, to having multiple phosphorylation event of the R domain, or eventually these are events which going back and forth the R domain?
Whether a single binding event...
multiple phosphorylation?
Yeah. Well, the truth is we don't know. we have no way to tell that. I would think that as long as the PKA is bound, it should be able to continue phosphorylating the various serines, but we have no evidence either against or for.
And apparently it have a
The affinity.
Yes.
So what we can measure is we can measure the apparent affinity for this reversible stimulation, right? And that is concentration dependent, and the apparent affinity is in the 100 nanomolar range, between 100 and 200 nanomolar approximately.
Thank you. Thanks again for your very interesting talk. So our next speaker is Catherine Olivier, and the title is Ribosome-Directed Small Molecules and Nucleotide-Based Approaches to Overcome Distinct Subclasses of CFTR Variants.
All right. Thank you very much, Gergely and Ines, for the opportunity to present today. And I'll just go ahead and mention the obvious, which is that I'm recovering from an illness last week. So you all get the baritone version of Catherine today. I'd like to just first disclose my relationship. It's a scientific collaboration with Ionis. And I'd like to cover 3 themes during today's presentation. The first is how we employ high-throughput yeast phenomics to identify genetic modifiers of mutant CFTR biogenesis, and I'll highlight ribosomal proteins as a few cases in point. Next, I'd like to make sure that we highlight the importance of translational velocity during CFTR synthesis, folding, and function, and this will be primarily illustrated by knocking down specific ribosomal proteins and evaluating those effects on CFTR biogenesis as well as the translatome. And then lastly, we will discuss the relevance of our findings to precision CF therapeutics, including ribosome-directed small molecules and antisense oligonucleotides that are presently under investigation. I'd like to begin by acknowledging the people who have done the majority of this work, which include 2 lab members in our group, Dr. Janice Jackson and Ms. Katie Foy, both of whom have posters at this meeting. So please be sure to swing by posters 330 and 287 to see their work. We also have former and current lab members who have contributed findings highlighted in this presentation. And then I've also been fortunate to work with a number of collaborators over the years who have played various roles in the data that you'll see today, and so I'll do my best to highlight each of those individuals as we go. And then, of course, thankful to our funding sources. Our goal really hinges on what I think most people don't need emphasizing at this meeting, which is that the majority of class 1 variants and other severe defects remain off-label for clinically approved modulators. And we also want to acknowledge that genotype-eligible patients, not all of them benefit from current modulators, and this can be for a number of different reasons that include adverse reactions or inaccessibility and more. So we have taken an approach to identify the mechanistic impact as well as the therapeutic potential of genetic modifiers that can influence the biogenesis of CFTR mutants that are represented in various subclasses shown here. And the way we've done that represents a longstanding approach with Dr. John Hartman at UAB, where we have modeled CFTR processing in yeast using the closest homologue the yeast express termed YOR1. This stands for yeast oligomycin resistance 1. So as the name implies, it's an oligomycin efflux pump. It's also an ABC subclass C transporter, just like CFTR. In the initial studies, we utilized the delta F variant in CFTR analogously expressed in YOR1, which is represented by delta F670. This variant exhibits features very similar to those observed for delta F CFTR, which include misfolding and an ER retention phenotype. And so as you can see here, the yeast expressing that YOR1 delta F are sensitive to oligomycin as compared to the wild type, which is resistant to this mitochondrial toxin. We have also gone on in more recent studies to model disease-causing variants in CFTR that are represented by the nonsense class of mutations. And so these are analogous, the G704X is analogous to G542X, and the L1286X is analogous to the W1282X in CFTR. And as you can see here, they are sensitive to oligomycin unless you add a read-through agent like G418 at increasing concentrations, which does restore the resistance and the functional expression of the YOR1. And so what John's group has done is employ synthetic genetic arrays to reveal genetic modifiers that, when suppressed, they can overcome the folding defects in delta F or other missense variants, or they could overcome even gating variants that have been studied, such as the G713D, that's analogous to G551D in CFTR. We've also identified gene deletions that can restore the processing of the nonsense variants as well. And so in all of these scenarios, we are endeavoring to identify oligomycin resistance that is increased or restored in haplodouble mutants that are generated from the studies. So this is just an example of how those yeast are replica-plated on growth media, and then growth media is supplemented with oligomycin. And so, as I mentioned, there's multiple subclasses of CFTR variants that we have studied to date and elucidated genetic modifiers of those variants. Specific genes that encode ribosomal proteins, initiation factors, tRNA processing enzymes, and more have emerged as robust hits from these screens. But what I'd like to spend time on today is really focusing on the deletion of RPL12 and RPL8 as one of the strongest results we've seen from these screens. With regard to the urine missense variants, RPL12 deletions seem to be a more robust hit, whereas for the nonsense variants, the RPL8 depletion seem to be more prominent in those results. So I'd like to just start with some foundational knowledge about RPL12 and RPL8 and where they reside in the human ribosome. This is a structure showing the large subunit in the light gray and the small subunit in the dark gray. The RPL12 protein is in the magenta, which you can see resides at the base of a flexible structure called the P stalk. This structure is known to interact with initiation and elongation factors during translation. But you'll also notice that RPL12 is residing within the GTPase-associated centers, so it will interact with any GTP-bound translation factor. What's particularly highlighted here is elongation factor 2 that's shown in the light blue. Now, RPL8 is in a different distinct region. It's embedded in the 60S subunit and interfaces with the 18S and the 25S rRNA, and this is near the E site of the peptidyl-transferase center. And so to summarize a large body of work that's been done over the years, I'd like to just highlight the way in which we translate the findings from the yeast system is to evaluate using siRNA-mediated knockdown the depletion of these genes. And I should note partial depletion. We usually endeavor to knock down these targets by anywhere from 30 to 50%, definitely no more than 70%. And we do this using a number of CF-relevant model systems that include Fisher rat thyroid cells, immortalized CF bronchial epithelia, as well as primary human airway epithelia. And so what's illustrated here are primary bronchial epithelia that are homozygous for delta F. And we have partially suppressed RPL12 using siRNA that's, you can see, only about 30% reduced. But what is interesting is that when we suppress RPL12, we can see maturation to PAN-C, as well as what you might get with a modulator like VX809 or VX661. And the combination of those two treatments does appear additive, if not synergistic. And so these data are quantified on the right with band C in the medium gray, band B in the dark black, and then the RPL12 levels in the light gray. And so you'll see more data like this in the rest of the presentation. And we also follow up these studies using functional assays like the Using chamber that's shown here, where wild type tracing is shown in the black. And then we have all of the delta F baseline versus delta F corrected with modulators or with the RPL12 suppression, as well as the combination thereof. And so I would just like to highlight that the knockdown of RPL12 seemed to rescue delta F transepithelial ion transport to about the same degree as what we saw with the correctors, but the combination restored delta F activity up to approximately 50% of wild type. So again, with this audience, I don't think I need to highlight that that is proposed to be clinically meaningful, being that it's above the 30% threshold that most people would suggest is what's needed to confer a benefit to patients. And before I move on, I would like to note that the way that the Using data are quantified in this figure and in subsequent figures is the forskolin response is shown in the black here. The potentiator response, like genistein or VX770, is shown in the medium gray on top. And then the inhibitor 172 response is shown in the light gray on the bottom. We have also gone on to test the effects of RPL12 suppression on other rare variants in CFTR. These have been stably expressed in Fisher rat thyroid cells. And so the one that's highlighted here is G85E, which is also a severe processing defect like delta F. You'll notice that there's no response of G85E to any of the modulators or the correctors like VX809 or 661, but there is a significant increase with RPL12 suppression. However, I will note that this is very modest because we only see about 20 microamps or so total activation, whereas for wild type CFTR in this setting, we see several hundred microamps. So this is maybe on the order of something like 5% of wild type that's restored. We also evaluated A455E, which is a class 5 variant, and we did find that knockdown of RPL12 did significantly enhance A455E activity, higher than what we have observed with the correctors, and the combination of these two interventions was additive. However, for P67L, which has both a conductance and a processing defect, we did not observe any beneficial effects of RPL12 suppression. So that was either alone or in combination with the correctors, where you can see just correctors alone give really robust rescue of P67L, but there was no further benefit from the RPL12 knockdown. And in collaboration with Zoya Ignatova at the University of Hamburg, we wanted to evaluate the effects of RPL12 suppression on the translatome. And so, again, I'm summarizing a large body of work that's already published that I would point you to this work, as well as the earlier work that was done with Gergely Lukac's group and Annika Brockman's group, as well as Eric Sorscher, where you can see that the effects were really nailed down in terms of, with single codon resolution, we performed RiboSeq to identify that RPL12 suppression is significantly slowing the rates of initiation and elongation, and this is primarily through effects on GC-rich codons. And so how that in turn affects the specific CFTR translational profile is the most significant slowing was observed in distinct subdomains that included the first membrane spanning domain, as well as the R domain and the second nucleotide binding domain. And so this mechanism overall, we hypothesize, is that we're allowing more time for the chaperones associated with the ribosome to achieve a more native-like conformation, which is improving the folding trajectory of this protein. But in more recent work, we were curious whether, because of the structural region of RPL12, whether it might be impacting other aspects of mRNA stability. And this is because earlier work from Roland Becker's group, is that they have shown RPL12 serves as part of the binding platform for the no-go decay machinery. And this complex is comprised of PILO in higher eukaryotes or DOM34 in yeast, as well as HBS1. And so we did a prolonged knockdown of RPL12 in CFBE, stably expressing delta F, and we found that around 72 hours is when we do see a significant, albeit small, increase in delta F mRNA expression. But really, at the 96-hour time point and beyond, there is a several-fold enhancement of these transcripts. So it's possible that we are interfering with the binding of the no-go decay complex to the ribosome without RPL12 there, but further detailed mechanistic studies would be required to evaluate if that's the case. So the interim points I'd like to summarize include that genetic interactions identified from the yeast delta F screen suggest genetic conservation between Saccharomyces cerevisiae and the human airway epithelium, and this is at least insofar as CFTR processing is concerned. In primary and immortalized human airway epithelia, RPL12 depletion reduces translation efficiency, stabilizes CFTR transcripts and protein, and promotes delta F functional correction, which we see in some cases is further augmented by CFTR modulators, but not across the board for some of the rare variants evaluated. So our present focus is to ascertain the impact of depleting gene targets like RPL12 and RPL8 and others that have been identified from yeast phenomic screens on the CFTR premature termination codons, or otherwise termed nonsense variants. And so to do this work, we initially constructed, with the help of Eric Sorscher's group and Zhang Hong and Andras Rob, FRT cells that stably express W1282X that encodes exon mini genes, or in other words, introns that are inserted around the exon affected, that's encoding the variant of interest. And so compared to wild-type CFTR, you can see that W1282X does produce a truncated band B and band C. When we suppress RPL12, we can see an amplifier type effect where there's more of that truncated band B, more of the truncated band C, possibly some mature band C, colocalizing here around the center point of that wild type band C. But in order to really evaluate if this was the case, we also generated these cells that encoded a C-terminal in-frame nanolute cassette as a measure of read-through. And so compared to G418, which is a very strong read-through agent, the knockdown of RPL12 was not nearly on the same level as what was observed with the G418, but it was significantly enhanced compared to the baseline. So this does suggest a modest degree of read-through. And then we also wanted to evaluate the effects of RPL12 suppression on the trafficking of 1282X. And so using a similar construct where the 1282X has an extracellular HRP tag introduced in the fourth extracellular loop, we evaluated the cell surface localization of W1282X in response to RPL12 knockdown and compared that to G418. And we can see that there was a very robust rescue of the cell surface localization. We wanted to take this next step and then evaluate RPL12 knockdown and its effects on 1282X function. And so using Using chamber studies, here's where we compared the knockdown of RPL12 on 1282X, evaluating in the context, again, of set to a wild type standard. And so the RPL12 suppression appeared to rescue 1282X activity to about 15% of wild type in CFBE cells stably overexpressing this variant. And we performed a similar study in primary human nasal epithelia homozygous for 1282X, where again we found knockdown of RPL12 was approximately 16% of wild type. In a separate set of studies, we evaluated the suppression of RPL8 and its effects on 1282X expression, but we also included in this initial test other hits that emerged from the UR1 screens that were encoding the nonsense variants. And so I'd like to highlight, again, the RPL12 knockdown as well as RPL8 suppression seemed to restore 1282X cell surface localization about to the same degree as what the read-through agent G418 performs. But there were other hits involved in the proteasome as well as tRNA processing enzymes that seem to confer a modest benefit as well to 1282X trafficking. We also evaluated RPL12 knockdown in combination with a number of other agents that target various aspects of translation efficiency, those that are both investigational clinical agents as well as those that are just used in vitro studies. And so here I'm showing, again, the same plasma membrane density assay for 1282X, and the knockdown of RPL12 alone rescues significantly the cell surface localization. However, when we combine RPL8 suppression together with G418, there seems to be an inhibitory effect, so there could potentially be overlapping or competing mechanisms there. But what was really fascinating is when combining the RPL8 knockdown with ETI, like acecaftor, tezacaftor, ivacaftor, we saw a robust enhancement of 1282X trafficking to the same degree as what we got with G418. So naturally, the next step was to evaluate these applications in FRT cells that, again, are stably expressing W1282X or wild type, and ascertaining the effects on function. So again, these are Using chamber studies. What was interesting is that just ETI alone seemed to enhance 1282X function to about 15% of wild type. But when we applied G418 or RPL12 knockdown alone, we did not seem to see any sort of benefit. However, there was a modest degree of additivity observed with RPL8 suppression and ETI, restoring 1282X activity up to about 19% of wild type. And so I think this points to the hypotheses about RPL12 versus RPL8 suppression having very different proposed mechanisms on the biogenesis of mutant CFTR. So returning again to our structure of the human ribosome, where RPL12 sits in this GTPase-associated center, we've got RiboSeq data as well as early mRNA findings that suggest knocking down this protein reduces translation rates and promotes a pro-folding type trajectory for misfolded delta F. But we've also found for a number of variants that there's increase in band B and band C and stabilization of some transcripts. So this would indicate that this suppression is exerting more of an amplifier type effect. In contrast, RPL8 depletion, we believe, might be conferring structural rearrangements to the 40S and 60S subunits, which could be reducing ribosome fidelity and so therefore acting in more of a read-through type capacity. And this is supported by earlier work performed in yeast that showed very similar findings. So it is proposed that they are having very differential effects. I would like to highlight, too, that we have done a number of off-target assessments on RPO12 knockdown because, as I mentioned earlier, we are very interested in taking these findings from yeast and turning them into therapeutic targets. So I'm running short on time, and I'll just highlight that we have done a number of assessments on cytotoxicity and have found that there's no evidence that this is the case in any of the CF cell models that we've worked with to date. We have also evaluated RPL12 silencing on the transcriptome and found that there are no global alterations to these profiles. We have also not observed any activation or repression of key cellular pathways that include the unfolded protein response or the integrated stress response. And we have also identified that RPL12 suppression does not exert global effects on proteomic profiles. And hopefully what I hope to be most of the convincing data is that we have found RPL12 depletion in vivo is well tolerated. And this was evidenced by an RPL12 heterozygous mouse line that we generated a number of years ago, where there were no detectable phenotypes that we could observe. And we also found that excised tissues from these mice, which are they're C57 black 6, and so expressing wild-type CFTR, wild-type mouse CFTR, we found that the processing of that protein was significantly enhanced in the pancreas, colon, and ileum. So how we've taken this as a next step into the therapy space, we have collaborated with Ionis Pharmaceuticals to validate 2 ASOs against RPL12, and we have found that either of these ASOs robustly enhance wild-type CFTR maturation as well as that of delta F. In more recent work, we've also found that delta F function is enhanced approximately 2 to 3-fold. But we, as they say, are not putting all of our eggs in one basket, and we're also attempting drug discovery approaches where in this setting we have FRT cells that are stably overexpressing RPL12 with a C-terminal nanoluke cassette. And this construct is just showing that we've got really robust expression of that nanoluciferase. Here we are evaluating through a high-throughput screening mechanism various known translational inhibitors, as well as a library of compounds that are provided by the Biological Discovery through Chemical Innovation Core at Emory. And so as an initial step, of course, we used all of the translational inhibitors that are familiar to the CF space, so ELX02, G418, PTC124, and none of those agents inhibited RPL12 suppression, but we were really surprised to find that eskin, which is a horse chestnut seed extract, does reduce RPL12 expression. We have gone on to further ascertain whether this effect is at the level of transcription, translation, or later in the processing of this protein, and at least with early mRNA data, it suggests that eskin is not inhibiting the transcription or the transcript abundance. So it would suggest it's impeding the level of at translation or later of RPL12 expression. So to summarize and talk about our future directions, we are endeavoring to elucidate the mechanisms by which silencing these specific ribosomal proteins overcome CFTR missense and or nonsense variants. We hypothesize there could be a number of pathways involved, such as inhibiting mRNA decay, peripheral stabilization of synthesized proteins, which is a known feature of 1282X CFTR, for example. We might be inducing read-through and more. I would also like to highlight we have additional gene targets that have been identified in distinct structural regions of the ribosome. Others that, besides the P stalk, they include the peptide exit tunnel as well as the GTPase-associated center. And so knocking down those proteins and seeing their effects on CFTR biogenesis. But we've also conducted subsequent screens with John's group to evaluate genetic or epistatic interactors with RPL12. So in other words, additional gene deletions that will enhance the RPL12 effect on mutant CFTR. We are also evaluating whether ribosomal proteins deplete, or whether ribosomal protein depletion rescues the CF phenotypes that are observed in CF mouse models, and this includes a collaboration with Craig Hodges. And we, of course, are continuing to pursue development and preclinical evaluation of anti-ribosomal ASOs and in small molecule inhibitors of these ribosomal proteins, and these studies are being conducted in vitro and in vivo through collaboration with Ionis and Craig. And so I've already done my acknowledgments, and I'd like to thank you all for your attention, and I'd be happy to take questions.
Thank you, Catherine, for this nice presentation. Any questions?
Hi. The inhibitory response that you got with the RPL8 knockdown with G418, you didn't see it with ELOX.
Correct. It was not observed with
So do you
ELOX-02.
have any idea why it would be different with one versus the other?
Yes. Well, I mean, my best guess is there's evidence that's been shown from Margarita's group and others that G418 does more than just read through. It also stabilizes transcripts. And so that's what we have found to be the same with RPL12 knockdown, with the RPL8, because it's more of a read through type agent. We haven't found evidence of mRNA stabilization, but that's also a possibility. We haven't dived into that mechanism as much as we have the RPL12 one. So yeah, in short, I think there's a lot of work to be done there. So we don't have a good answer yet, but it's a great question.
Thank you for a great talk. I just have a couple of questions on the mechanism. So you're talking about two proteins that are one, a core ribosomal protein and the P stalk, right? And so what are your thoughts about how the depletion of these would affect ribosome synthesis in the first place? So even though ribosome is the reader of these nonsense messages or identifier, but at the same time, it's also required for translation of your transcript in the first place, right? So how, with this depletion, do you balance the two?
Mm-hmm. That's a great question that I get all the time. So the first thing I would mention is that, again, we're trying to tune the knockdown. We're not trying to completely ablate these proteins. So I do think, like any therapeutic approach, you're going to have to find that effective window where you're seeing a benefit to CFTR correction, but not at the expense of cell cytotoxicity. And so when we aim for about 50% reduction in these ribosomal proteins, that's across the board. We've never seen any deleterious effects like that. But I will also mention that we want to be cognizant of loss of function of these proteins being tied to any other clinical manifestations. And so ribosomopathies such as diamond black fan anemia are well known. RPL8 loss of function mutations are associated with that disease. However, RPL12 has not been implicated to date. So it is something that we are cognizant of in the back of our minds. Maybe the other thing to mention is that when we're doing these partial knockdowns of ribosomal proteins, it's presumed that we've got different subpopulations of ribosomes. So it could be, again, we're not causing a deleterious effect on the core translation that's happening for all proteins across the cells. But there may be something that's relatively specific to the expression of CFTR here that would be suggested by some co-IP studies we've done where we have found direct protein-protein interaction with RPL12 and CFTR. But that hasn't been done yet with RPL8.
Thank you.
Mm-hmm.
That was a great talk, Catherine. Thank you. My question is, did you try additivity or synergy of RPL12 knockdown and ETI? Because you showed with VX809, but not ETI.
Yes, we have performed that on delta F as well as W1282X. And in general, yes, there is additivity. It seems that it's more synergistic effects with RPL12 and ETI for delta F, but for 1282X, it's more in the additive range. We only see a slight increase, maybe a 3 to 5% bump there.
Alexa alone?
Have not done Alexa alone.
Okay. And the other question is, did you have a chance of looking into polysome profiles?
Yes. This also feeds into the earlier question too.
Exactly.
So yes, we have done global polysome profiles with the RPL12 suppression, and we did find that there was a reduction in the total abundance of the ADS monosome as well as the 60S subunits, and then all of the polysomes. So that would indicate that we might be abrogating an aspect of ribosome biogenesis, but again, it's not enough to cause widespread havoc on the cells. We have only observed a 20% reduction in cell growth. And I do think that there's also literature out there that suggests if you can reduce initiation rates of ribosomes, then you can actually restore or increase the expression of genes that are subject to mRNA decay pathways, such as no-go. So that would be the hypothesis, is maybe that we are abrogating a little bit of ribosome biogenesis, but also decreasing initiation rates to then overcome the expression defect of 1282X in particular.
Right.
But maybe
Good
other
hypothesis. Yeah.
Yeah.
Yeah, really nice talk. I have a couple of related questions. So first of all, RPL 12 and 8, I'm guessing are probably put on the ribosome in the nucleus before, not in the cytosol.
RPL8 is, RPL12 is in the cytosol.
Okay. So the question is the cell trying to repair the ribosome, or is it possible to add these proteins back post-synthesis? And if not, is the cell destroying these ribosomes? Is there a quality control process that's recognizing, oh, this is an aberrant ribosome and it's going to be turned over more rapidly? Have you looked at turnover?
That is a great point, and no, we have not. But yes, I would love to do that.
Thanks.
Mm-hmm.
Okay, so if there are no further questions, we can come to our next speaker, who is Gary Cotting.
Thank you.
So our next speaker is Gary Cutting, and the title is Finding from the CF Genome Project that Informs CF Pathophysiology.
Got it. Thank you. Okay. Thanks. Well, it's 5 o'clock. It's 8 o'clock in my brain from the East Coast. But thanks for hanging around. My talk's going to be a little bit different. We're going to talk about a resource that's available to you, and I'm going to basically just skim across a few findings we've had so far. There's 12 manuscripts that have been prepared, 5 published already, and there's 7 published already, 5 in process, and my collaborators have generously allowed me to include some of that unpublished information in this presentation. So this genome project is basically started about 4 years ago to deep sequence about 5,000 patients with CF. And so I'm just really presenting on behalf of our consortium the information. But again, I can't emphasize enough about how much this is a rich resource for you if you want to dig into the genome to ask questions like, do variants in RPL12 or RPL8, as Catherine just presented, in some way alter or modify CF disease, or any other gene you have, because we sequence the entire genome. So with this, I'll just mention I have no disclosures. This is the group that's worked together, and members of the Johns Hopkins University group, myself, Scott Blackman's the other PI, and a large group of other analysts and so forth. University of Washington, Mike Bamshed is led up from one group, and Ron Gibson from the Seattle Children's Hospital. UNC has been another collaborative group with Mike Knowles and Wanda O'Neill. Wanda's actually just been presenting some of their results in a separate symposium. And also North Carolina State University with Fred White and his group have contributed. The CF Foundation has been an important partner through this whole thing. Chris Pendleton initiated the project, discussion with Bill Skach and others, and Martin Mentz, and also assistance from analysts at the Foundation Therapeutic Lab, Audrey Shevchenko and Norman Allaire. It's supported by 3 grants from the CF Foundation and also from funds supported by NHLBI through their Biodata Catalyst program. This information, again, I want to emphasize, is available to you. You can directly apply to the CF Foundation to Catherine Tuggle to get information from this genome sequencing and the clinical data. Or if you wish and you'd like to work with any one of these groups, please contact us because we're happy to collaborate with you. So just to give you a little bit of a description of the project. So this is paired whole genome sequencing. and a harmonized phenotype data set. Harmonized means that we've actually worked to get consistency in the phenotypes across these 3 different study groups, because they're composed of about 1,900 individuals recruited by Hopkins who are actually twins and siblings with CF, and also individuals with CF-related diabetes. There's about 1,800 from North Carolina, and they're from the gene modifier study, and also a subset that have severe liver disease. University of Washington has utilized their recruitment from the EPIC study, focused on Pseudomonas and other infections. So there's strengths of each of these programs, and the kind of clinical data that was collected, extracted mostly from the patient registry that the foundation maintains, has been, again, harmonized across the groups because, to start out with, there's different ages of the patients in this group, as you can see here. Younger patients from the University of Washington group, older patients from UNC, and then Hopkins was in between. So already with those age shifts, you've got to work on, again, harmonizing the data, because obviously FEV1 and things like that are lung measures have to be harmonized. The actual sequencing, so 120 million variants were obtained, and about 105 million of those were then passing QC, and then filtered down to finally about 89.5 million variants are in that resource that have been quality controlled and checked. There were 71 duplicate sets in there. We didn't actually know about some of the duplicates, were recruited more than once by one study and the other study. We were able to reconcile that and show that, in fact, the genome sequencing is quite consistent amongst those 2. There were 10 pairs, maybe 20 actually now, I think, of MZ twins in the set from Hopkins. Another way of checking that you get the same sequence information, because you expect the MZ twins to be identical, and they were. So let's talk about what kind of information. Again, I'm just skimming really so far what we've done in the time that I have. What have we learned from this? So let's first talk about CFTR, since that seems to be very important in CF, and we've heard quite a bit already about the protein. But what about that gene and its variants? And we're still trying to resolve this issue of finding every variant in every patient with CF. And I mentioned to you that currently most clinical sequencing or sequencing people do of this gene involves just the coding regions. So what I'm showing you above, if I can, these are the regions that are currently covered, just these small regions that are the exons, the coding for the protein, and the flanking introns. There's large regions that are not covered, but of course, if you go ahead and sequence entire genes like we did, which CFTR is only just one of them, you get the whole thing, basically. There's a few gaps in here due to repeat sequences, but in general, the entire gene is pretty much covered, as well as you can now look at flanking sequence on both sides of 5' and 3' regions, where you're hoping to see sequences that regulate the expression of the gene. So what happens if you look at the whole gene? What do you get? What's the kind of improvements in finding variants? Well, you can find things like structural variants that are not found. Many of them involved introns and exons, and are difficult to find by the other forms of sequencing that are done. And you can find quite a few of these intronic variants. Intronic variants change the splicing of the gene. And in fact, it's important to find these kinds of these other variants, because the therapies change. Structural variants, which I'll show you a list of them here, is already published, are going to require replacement of the gene or replacement of chunks of the gene. Intronic variants, which are usually point mutations, are going to alter the splicing, and you can suppress some of these by using oligonucleotides. And we've seen examples of those therapies here at this meeting for the 3849 gene variant. And so therapy implications are important here. Also, you can complete the genotypes in these individuals. So if you have to look at these 5,000 individuals who underwent this study, in this first phase analysis, 2 CF-causing variants were identified in 5,011 out of the 5,058, so 99%. So we didn't do any further analysis, and this was published. Less than 2 CF-causing variants were identified in a very small fraction, less than 1% actually, 47. And then the question was, well, now we've done whole genome sequencing, and we can only find 1 CF-causing variant or no CF-causing variants. Is there some other answer elsewhere in the genome? And so a second phase analysis was done. It was really an interrogation of the genes, and I've just summarized the information here below. But 545 loss of function variants are found in almost 500 genes. And in that situation, there were deleterious variants found in 3 people, 1 gene each, 2 genes that are involved in primary cellular dyskinesia. One is carbonic anhydrase. Our lab had reported that variants in that gene can cause a CF-like phenotype years ago. But despite that extensive share, none of these individuals had a variant in the second gene. So they really only had a single variant. So they're probably just carriers of a change in these genes. And so we couldn't really resolve them with this thing. So we're, at this point, despite having the genome sequence available, we could resolve these cases. But we're left with the rest of them. No deleterious variants found in 44 subjects. And so we thought, well, what is going on? So let's go back to the phenotype. And actually did a phenotype review and discovered, after looking at these in detail, and these people recruited as part of a modifier study for cystic fibrosis. So we assumed they had CF. But you got to be careful when you assume things, because when you look back upon this, you find that 43 of them really we couldn't confirm the diagnosis. So what does this tell us? Well, if you go ahead and sequence the entire CFTR gene, and you've looked very carefully, and you can't find 2 CF-causing variants, they probably don't have CF. You'd think that's kind of obvious to us, but, you know, now we're starting to realize that, you know, genetic investigation has some value, and particularly in the age of modulators, you got to be careful about the patient really having CF or not. So anyway, so it's a good assay both for finding the variants when they really are causing CF, and CFTR dysfunction is the pathophysiology of the disease, and also excluding CFTR when it isn't. Unfortunately, the whole genome sequencing for our point of view was a little disappointing because we didn't find other causes, genetic causes at least. So what about those small number that have just one disease-causing variant? Well, maybe there's another variant in there that we just can't interpret. That's quite possible. Could be a symptomatic CF carrier, and there's been a lot of talk about whether or not individuals who are haploinsufficient for CFTR have just one functioning copy. Maybe that's not enough in certain cases. There may be epigenetic explanations, and we start to think about how to go after that, such as changes in methylation or chromatin alterations. Or it could be what we in genetics call a phenocopy. That means it looks like CF, but it's a mimic, and it's not due to genetic causes. So let me now switch gears and from CFTR to now think about what type of genes we could uncover that would contribute to certain aspects of lung disease. I'm going to go through 2 aspects. The first is airflow, and the second being infection. So let me focus on the airflow. Much of this work was done by, at least led by our colleagues at UNC, and it's based on something we developed some years ago to, again, harmonize the lung phenotype across all of these patients of different ages, which is this quantitative Coolidge normal residual mortality adjusted measure, which is based on FEV1. So the study was done where we combined data from the CF Genome Project, those 5,000 individuals, and added to that were individuals recruited by our colleagues in Canada and in France, and they're part of the international group here, and I've indicated our symbol up there. So this was a very nice opportunity for collation of data from several sources to bring together one of the highest or the largest GWAS type studies that have been done in a single gene disorder. And so we've got almost 8,000 individuals in this study, and they're all pre-modulator. So you can imagine what's going to happen now if you want to try to do studies looking for modification of CF. You're going to, in this day and age, is you're going to have to consider the effect of those modulators. This resource will give you information pre-modulator. So you can start looking to see if genes were contributing to variation in lung function in this data set, and ask the question, does the advent of modulators shift the effect of those genes or not? So we could start thinking about how modulators might be altering the way in which genes modify this trait, this pulmonary trait. Now, this is a a Manhattan plot. I think you guys are familiar with it. I guess I'm subjecting you to this because as a geneticist, I got to see all those wiggly lines that the physiologists showed me for years. And I finally got to figure out most of them, as well as the structures to some extent. But this is a plot of all the chromosomes and all the variants. And there's several million variants. You can't see them all. They're just all plotted across here, and they're plotted against the log p value. That log p value is telling you which each individual variant, whether it associates with the trait that we're working with. So I'm going to show you this over and over again. And it's just, this is the format that we use within genetics. So if it does associate, and if it passes a rigorous threshold for significance, which is the number of tests you're actually doing based on the number of SNPs you're using, then you can start really thinking it's really associated. It's really a signal. Now, if I put CFTR in here, it would be through the roof. You can probably find CFTR nowadays, well, you can find it by whole genome sequencing in a couple of patients. If I do one, find delta F and you're done. But because that's what we can do nowadays in a clinical lab. You can bring somebody in with a particular disease, sequence their whole genome, and you can figure it out with that patient. If you get a second patient with the same phenotype, variants of the same gene, now you're pretty sure you've got the right gene for the disorder. You do a third one, statistically, it's been demonstrated that you have a high degree of confidence you have the right gene. So 3 people, nowadays, you could solve or discover CFTR as the cause. So that's the power of this type of technology. It's also very powerful of linking variants from different genes and loci with traits, and such as in this case. And here we have a couple of them, and I'm going to focus on MUC4, MUC20, and this AGTR2 locus. Here's the MUC4, MUC20 locus. The SNPs are highly associated. I can show you here. This is in the 10 to the minus 11th range for the p-value, and it's right on top of the MUC4 gene. These are tethered mucins, and there's MUC20, and these tethered mucins, you can see just about on this study that was published, the tethered mucins with the arrows here are shown by this EM now attached to the cilia, and they play an important role. Here's this is not a pic, this is a diagram of this. And here are the tethered mucins for MUC4 in orange and the MUC20 in purple. The cilia are shown of these airway cells here, and they extend out in this way here. And what we found is genetic evidence that somehow variation in the expression level of these genes, probably MUC4, but it could be MUC20. It might be something else in this region, but these are the best candidates, are altering this FEV1, the airway measure that we're using here. So what about this other locus, AGTR? So this one is a cluster of SNPs right on top of AGTR, very beautifully lining around this. And then Mitch John with Rebecca Dara did this study a couple of years ago that with genetically modified mice, they knocked out this AGTR, which is a type 2 angiotensin II receptor gene, and it normalized pulmonary function indices in 2 independent mouse models. And they also use pharmacological antagonism of AGTR2 to improve pulmonary function. Those mice are near wild-type levels. So we actually have alternative approaches to modify lung disease and CF beyond going after using CFTR modulators. So the question is, do we have the resources and the interest to start actually taking these studies and thinking of applying them in humans? The other things that came out of this study, which is really, I think, very interesting, is once you have this rich resource of data and genes, you can start looking not just at individual loci, but clusters and genes that are found in the same pathways. And a beautiful work that was done by the UNC group was then identify genes that are cluster in branching morphogenesis pathways. This is the development of the airway branching, and I won't show exactly how this is done, but just this is the data in their paper that's recently published, and this is basically showing all of these genes of significance are enriched in these pathways, and they had just put a cutoff right here. But what I'm showing you is the genes that they listed underneath. All of these are significantly associated as a group with variation in airway flow in patients with CF. And they outlined 3 pathways, the hedgehog, sonic hedgehog, TGF-beta, and the Wnt pathway. So what this kind of mining is telling you is that genes in certain pathways, and you can do it for a variety of ones, but in this case, pathways important for lung development are, and it's no surprise, are important for airway function in CF. And their variation in these genes influence the severity of lung disease in patients with CF. But that's a lot of genes to start working on. But it's at least instructive from the pathophysiology point of view. Pseudomonas aeruginosa. Now, we've heard that it continues to be a big problem in CF, even in the modulator era, and we want to find new targets. Bacteriophages are very interesting, very helpful perhaps to go after the Pseudomonas, but what else can we learn from humans? And studies done by our colleagues, led by our colleagues in Seattle, looking at chronic Pseudomonas infection. Again, looking at GWAS, and here these are locus zoom plots. This is right on top of the, zooming on the region where we've found association. And you can see that there's a very nice signal here, at almost 10 to the minus 8, right on top of this CHP2 gene. In fact, 2 copies of the minor allele of this variant has a 3-year difference in age chronic Pseudomonas. So whatever this region is doing, or potentially this gene, is quite interesting. Now, this gene encodes this calcineurin B homolog protein 2, binds to NHE1. It's important for intracellular trafficking and control of intracellular pH I mentioned. It's got some T cell functions as well. Others are working on it, but the key is, what is it doing in CF? Okay. And so, but at least it's a reasonable candidate to start thinking about for variation in Pseudomonas infection. And it provides us as a new way of thinking about this disease from a different direction, potentially therapeutic targets. NTM is another challenging problem in CF. You've probably heard about it several times. It made up of a couple of different disorders, but the 2 ones that are most often seen are Mycobacterial avium complex, MAC, and M. abscessus. And both of these contribute to this NTM condition. When you get NTM and you have several sputum cultures positive for it, it's associated with greater FEV1 decline, as noted here. Study done again by our Seattle colleagues with nearly 2,000 individuals with no NTM against about a very, very small group of individuals that had NTM with 2 positive MAC cultures, showed a very nice hit on chromosome 2. And this is right over top of, or very near, this ICOS gene. Why is this ICOS gene important? Well, ICOS has already been actually implicated in TB infection in the general population. Okay, so this is a theme you're going to see over and over again, is that some of the genes that are implicated as modifying CF are also playing roles in the pathophysiology of similar or the same phenotypes in the general population. So every time we find these genes, we can learn from our colleagues what they've already studied in similar phenotypes. Intestinal obstruction and diabetes. This is work that the Hopkins group has taken the lead on. So here's 2 traits. Just keeping my own time here. Here's 2 traits that appear to be somewhat different. Intestinal obstruction is an in utero problem, and there's a baby that's just been born with this, with all of these dilated loops because the obstruction of the ileocecal junction, and well described. We can treat it, surgery or medicine. Diabetes, it's primarily due to problems with the production of insulin from the beta cells within the endocrine pancreas. As we all know, the exocrine pancreas is the major problem in CF, and we believe that inflammation of the exocrine pancreas leads to problems with these islet cells and reduction in insulin. This has a age of onset that's progressive over time. The 2nd to 3rd decades, diabetes starts appearing, and if patients live into their 50s and 60s with CF, they'll all get diabetes. So there's 2 very different complications of CF: intestinal obstruction, an in utero problem, and diabetes, an age-related complication. So here's the GWAS studies that were done on those 2 traits. Here's the intestinal obstruction. I point out ATP12A. Mike Welsh and his colleagues have indicated this non-gastric proton potassium adenosine triphosphatase acidifies airway surface liquid, and it impairs host defenses. I will point out non-gastric. Well, it's coming up in intestinal obstruction here. What the heck's going on? So maybe there's things these genes and proteins are doing in tissues we didn't realize or we didn't get right. Or they're doing more than one thing. They are doing, indeed, as Mike and his team has worked out, and others, in the airways, but they're playing other roles in the intestine or somewhere else that are contributing to meconium ileus. Diabetes, TCF7L2. TCF7L2 was found associated with type 2 diabetes about 8 years ago. It has been replicated over and over again in over 100 studies of patients with type 2 diabetes. And it is a risk factor for diabetes and CF, without question. And we know that diabetes is not really that well treated with the modulators so far. So we do need other targets to start thinking about. And the amount of effort that's been put in TCF7L2 is massive. Okay, I better move on. So there's two other loci, CEBP, that is found in both traits and is associated with type 2 diabetes. This SLC26A9 is found expressed in the exocrine pancreas and in the lung. And the story is quite interesting with the lung, and I will not go into that right now, but I'll just talk about the pancreas side. This trait is called pleiotropy in genetics. This is where variants of one locus or one gene influence more than one trait. So obviously there's something going on that is the genetic architecture is similar between that intestinal obstruction and diabetes that we didn't realize before. What I'm showing here is now the loci that I mentioned, the 26A9 in common, and when we deep sequence, we found another locus in this region. There's something else going on, more than just the 26A9. And when you link the variants together, you can see that when you put the two loci together, you can range from 22% affected with diabetes up to 40%. So there's more than one locus operating in this region on chromosome 1, the A9, and this A3 locus. And when you put the two together, it doubles the risk for getting diabetes. So what do we think is going on? Yep, I'll keep moving here. We believe the following, that there is a series of variants associated with risk for diabetes sitting just 5' of A9 in the promoter region. Our lab, Onto Lam from my group, had done reporter studies with this in 2 pancreatic cell lines and demonstrated clearly these variants alter the expression of the reporter gene here. There's also evidence done from GTEx, another resource that shows variants in that region alter the level of expression of SLC26A9. So there's pretty good evidence that 26A9 is doing something, and it's expressed in the pancreas and the pancreatic exocrine cells. So it looks like that those variants are probably operating on A9. But EQTL data also shows that these variants operate on the level of expression of this adjacent gene called PM20D1. And interesting enough, the other locus is not operating on A3. The EQTL data says it's actually varying the level of expression of PM20D1. So when two things point to the same gene, you usually want to pay attention to it. So it's probably doing something in this case. And the interesting thing is the commonality is PM20D1 is contributing to the diabetes phenotype, and the A9 is contributing to the diabetes and the MI phenotypes. What do you think is going on? With D1 is in fact expressed. This is tabulus sapiens. I don't know if any people are familiar with this. This is entire tissues from a human. CFTR is found in the pancreas and in the lungs, pretty much, not too many other places, believe it or not. And PM20D1 is found in the pancreas. What does the pancreas data look like? Well, here's just a single cell analysis. These are the pancreatic acinar cells. These are the ductal cells. These are the components of the islets. And CFTR is found pretty much in the pancreatic ductal cells, as you expect. PM20D1 overlaps to some degree and is found in the acinar cells. So if you lose your acinar cells, because you have pancreatic exocrine insufficiency, you're not only going to lose CFTR function, you're going to lose expression of PM20D1. And what is PM20D1? Just because of time, I'll flip through this. PM20D1 is involved in glucose homeostasis by the fact that it plays an important role in the formation of N-acyl amino acids, and it's important for the conversion of brown fat into regular peripheral fat and backwards and forwards. So there's a whole new story that's evolving from other investigators as to this new way in which this protein and others, by altering fat metabolism, are contributing to glucose homeostasis and also insulin expression at the end of the day, because this seems to be linked now to diabetes. So a completely new pathway we didn't expect, express protein in the pancreas, and so now you can actually test for levels of this protein in patients with CF and those with and without diabetes. I'll finish here with body mass index. FTO, I just point out, is one of the most highly associated regions with body mass index in the general population, and it's playing a role in CF. But there's a new locus, ADAMTS5, that we discovered that pretty much is only found in patients with CF, and it plays a role in the development of brown adipose tissue and browning of white adipose tissue. So we're seeing a theme evolving now, because now we've got body mass index also showing evidence that things that alter the peripheral fat and its status are playing a role in modifying body mass index. And so we think of selective targeting. This is actually specifically from this paper in Metabolism. Selective targeting of ADAMTS5 could provide a novel therapeutic study for treatment, prevention of obesity and metabolic diseases. The final point is we never would have found ADAMTS5 if we did not include patients who were of African ancestry, because Africans have a more diverse genome, because they're more ancient and they carry more variants. And we were able to see that out of the white patients, only 17 carried this particular variant. And that was 17 out of 3,183. Here, out of the total 17, and there were 73 that didn't carry it, but a much higher fraction, 20% of them, carried this variant. So therefore, we're able to detect it because we included these patients. So another important lesson we're learning is the importance of including diverse populations when you do these kinds of studies. So insights derived to date. Full CFTR gene analysis of just over 5,000 individuals. The vast majority of CF is due to defects in CFTR. That's no surprise to you. But a negative test is quite useful. Go back and check the phenotype if you're coming up with a negative full gene sequence. In-depth analysis of loci associated with lung disease severity. Prioritize, as I pointed out, modified genes, and I mentioned MUC4, perhaps an AGTR. Implicates pathways involved in lung development and repair. Also novel targets for Pseudomonas and NTM. I talked about pleiotropic effects, the way gene and gene loci can affect more than one trait, and how we can dissect out the pathophysiology of those disorders. And finally, variation body mass index and the fact that use of diverse population are highly informative. Where can you go next, or with other things? Well, we're in the post-modulator era, and here's a waterfall plot. Now, every patient responds the same way to the modulators. So you can go back in and now collecting the data post-modulator and compare pre-modulator, post-modulating, so asking questions of this study set. What changes? What loci change? Which ones are modulator resistant? Because if they're not altered by the modulator, they might provide a new pathway and a new target to go after to complement what we're doing with the modulators to treat not just CFTR, but to treat the other manifestations of defective CFTR in the lungs and in the pancreas and so forth. There's also this huge wave in genetics of using something called a polygenic risk score. And it's made up of, again, variants from the general population that show risk for certain traits, and then you can actually use that information as predictive. So if a certain person has in them a number of variants that are risk variants, that combination of risk variants changes the likelihood you're going to have a disease, or you're going to be one end of a BMI spectrum or others. And this is going to be probably, you'll see this used more and more in your lifetimes, and you're going to get tested using these polygenic risk scores to now stratify you according to your risk. We can apply the same things in CF, and we think this could be done. My final point, and thanks to my collaborators who've allowed me to present on their behalf, a little bit too long, but this resource is available. So please, if you want to use it directly, you can apply to Catherine Tuggle within the foundation. You'll have the information on the phenotypes. You have the information on the genotypes. If you would like some help with it, we're happy to help you out. If you'd like more help with it and really want to get involved with us or help us with your project, just let us know. We'll be more than happy to collaborate with you. Thank you for your patience for my slightly overlong presentation.
Thank you very much, Gary, for this very interesting update on the CF Genome Project. Margarita has a question, as I can see.
That's great, Gary. This gives us like 10 years of work to find out what's going on with this.
10 years of work for you, Margarita. I want to retire.
Anyway, for the 44 patients, which initially you didn't find the 2 mutations, so what were the inclusion criteria? So how were their sweat chloride and
well, in a number
why
of cases,
were they included?
Yeah, well, they were supposed to have sweat chloride above 60. They were supposed to have clinical features of CF, and they were supposed to be enrolled by a physician familiar with the disease.
Right.
But, you know, sometimes I'm a physician, and I'll just admit, we don't always get it quite right. And sometimes the sweat is elevated due to inaccuracy in the actual assay itself. Sometimes, you know, the phenotype, it looks like CF, but isn't really. And so I'm pointing out that, you know, if the error rate is, say, 0.5% in what we're doing in defining patients with CF, that's not bad, actually, because if you look at our error rate in other diseases where we diagnose, it's usually considerably higher than that. So I think it's part of the noise in the system that's there, and we can only assume that the information used by the clinician to define it as CF was accurate at that time. They've obviously gone back to reflect that that information probably is not correct, or reevaluate the genotype and realize the patient has, reevaluate the patient and realize that that's not the diagnosis anymore, or we couldn't confirm it because they're no longer in the CF patient registry. They disappeared.
I guess that a fraction of those actually had 1 mutation, right?
Nope.
Zero.
Well, some of them did. Yes, some had a fraction. So there were probably carriers that somehow got into the system, and one could go back and think, is that what's going on? So, but since you can't phenotype it, you can't confirm the diagnosis of CF. Now you wonder, well, is this a CFTR related disease? Which is possible. So it could be that CFTR with 1 defect, being a carrier with 1 loss of function, maybe they've got some CF-like disease, maybe monosymptomatic, and maybe that's how they got into the study. Yep.
And I was actually wondering, you know, in the final part when you mentioned this polygenic score, whether these would be candidates, because maybe a number of unfavorable variants could actually make it if you are already a carrier. So.
Yeah, exactly. So the combination of a carrier status plus a combination of other things may be enough to tip you over into the disease. And then again, if that's indeed what those genes are doing, because there's variation in expression or function, then of course you got to think, are they therapeutic targets? And they very well could be.
Yeah, thank you.
Hi, I have a lot of interest in genetic modifiers of modulators, and I was wondering, are you tracking these patients and their responses to modulators or any other outcomes over time, or is this just you just collected the initial data?
Well, we are going to collect a cross-section. We really would like to do a longitudinal study, and that's really the way to do this. Okay. And if anyone want to help me convince the CF Foundation to spend another few $1 million to track patients, that really would be the way to do it. But I believe our group has the way now. We have new information from the registry that is post-modulator. So we now have a data set, and all the information I showed you was pre-modulator. We now can compare post-modulator and start asking ourselves, what's happened to those GWAS studies? Are the same loci coming up, or a new loci now appearing? And what's in those loci? And maybe there's something we can go after. Yep.
Okay, thank you.
Hi, I just couldn't help thinking about, I studied ribosomes for most of my research. And so there is a similar situation where in Shwachman-Diamond syndrome, where a particular protein was assigned the positive agent, but then it was part of a pathway. And then later, a lot of different proteins that were contributing to the same biogenesis pathway of the large subunit resulted in similar situation. So one thing I was thinking about earlier, we heard a wonderful talk about a PKA that modifies CFTR and regulates its activity. So one could think if that metastylation, which associates pKa with the membrane, doesn't happen, wouldn't that affect CFTR activity? And similar such things that contribute to CFTR regulation could be.
Well, since we have the world's expert over here, but I won't even, all I can say is the following. If it just happens that there are genetic variants, just by chance, this is a bit of a chance thing because there are genetic variants in a gene like that encodes pKa. Okay, and those genetic variants are there. You can then, we know there are variants, but if they actually change the level of expression or they change the function, you may have the opportunity now to take those variants and run them against these phenotypes in the CF patients and ask, do they change the phenotype? If they don't, it doesn't exclude that pKa is important. It does not. But it gives you evidence, okay, that if you do find some kind of association, that protein is important. Now, we know pKa is important. But other ones that you're not so sure about, you're working on, you found something interesting, a cell biological study, and it's a chaperone that interacts with CFTR, you can ask the question of this data set, can I dive in there, find genetic variants in that gene, and can I then run that against the phenotypes that I've just mentioned, there's others you may want to derive from the data set, and say, does it modify the disease? If it does, then it provides positive, useful information. If it doesn't, it doesn't exclude anything. So it's kind of a great experiment to do. It's a no risk, right? It's if you win and there's an association, great. The only thing you got to do statistically is be very careful about the p-values you use and the number of comparisons you make. Okay, but that's why the resource is useful. It's got 20,000 genes in there, and if you guys come up with a protein, maybe you want to take a look at the gene and its variants and its association with phenotypes in this data set.
Thank you.
Thanks again for your patience.